Hello everyone, myself Hemant Malekar, Department of Botany, Anand Niketan College, Anandwan Warora, District Chandrapur. Today I am delivering my presentation. My presentation topic is Melisopylenological study in some honey sample from Vani Tahsil, District Yavatmal, Maharashtra. My presentation contains introduction, review literature, methods and materials, observation and result, discussion and conclusion, scope of this study and references. Introduction. Palynology is the study of pollen grain produced by the seed plant and spore produced by pteridophyte, bryophyte, algae, fungi. The various branches of palynology are aeropalynology, melisopylenology, paleopalynology, and forensic palynology. In recent years, melisopylenological studies attained a global status. Melisopylenology is concerned with the identification of pollen in honey. A pollen analytical study of honey provides a basis for identifying the grains of honey in terms of locality and floral sources. The pollen analysis of honey sample is the best method to determine the plants used by bees as a source of a nectar and pollen. The honey is a sweet food made by bees using nectar from flowers. Honey is the most innocent natural sweet substance appeared on earth. Honey is the most familiar food and used in the various food products like jams, jellies, cakes, bakery products. It contains all essential nutrients rich in carbohydrate, vitamins, amino acids, proteins and minerals. The variety produced by honey bees is the one of the most commonly referred to and is the type of honey collected by beekeepers and consumed by humans. Honey produced by other bees and insect and distinctly different properties. The beginning of a microscopical examination of honey go back to the year 1885 when Pfister exam by the pollen analysis is the number of Swiss, French and extra-European honey and demonstrated the possibility of determining the geographical origin by the pollen grain contained therein. In identifying the pollen grain, he made a use the work of Fisher 1819 and Firiget's 1837 and Mohal 1834 on the structure and morphology of pollen. Materials and methods. Study area and region. The total 8 honey samples were collected from tribal people of different villages in Vani Tahsil district Yavatmal during the month of August to December 2019 and assessed by using the authentic melisopylenological methods. The honey sample were classified as a sample 1 Bhallar, sample 2 Lathi, sample 3 Shirpur, sample 4 Besa, sample 5 Kundra, sample 6 Purad and sample 7 8 Ganeshpur, sample 8 Nilapur. The collected honey was stored in plastic bottles and refrigerator at 20 degrees Celsius to 25 degrees Celsius. The analysis of honey sample was done in the laboratory in the accordance with the known method, the Lewinox and Suryanarayana. The sample preparation, the pollen analysis of the honey sample were carried out by dilution method. The dilution were prepared from each honey sample by taking 10 gram of honey in a 10 ml warm distilled water and centrifuge. The pollen grains were prepared for light microscopy by the standard method described by Edmund and Aurora and Modi. For light microscopy, the pollen grain were mounted in stained glycerin jelly and observation were made with a trinacular fluorescence microscope. The measurements are based on 10 readings from each pollen type and the pollen grain size, colpi size and were most measured. The terminology used: the list of plant species found in the analysis, pollen in the honey sample, uh, the allium cepa, Altanthera sessilis, Azardicta indica, Brassica nigra, Carya arbora, Cassia tora, Cassia samia, Butea monosperma also. This presentation only for uh, qualitative analysis, the parameter study. The qualitative analysis were carried out by the method described by Leeuwenhoek. The pollen percentage for each pollen type was calculated and the pollen frequency and frequency class were measured. The honey was classified as either unifloral or multifloral on the basis of pollen count. All the studied honey sample was found to be a multifloral. Sample 1 is a multifloral. The absolute pollen count in 249 was noted. Sample 2 is the multifloral. Sample 3, all the samples are multifloral. This is the qualitative analysis of honey sample. Sample 1, sample 2, sample 3, sample 4, sample 5, sample 6, sample 7, sample 8. This is the observation and images for result. First one image is the Altanthera sessilis, second is the Allium cepa, Azardicta indica, Brassica nigra, Butea monosperma, 6 is the Sina samia, Sina tora, 8 is the Carya arbora and Delonix regia. Total 18 pollen was studies in this work. Discussion and conclusion. The pollen grains found to be an essential tool in the analysis of honey. In the present investigation, it was observed that the morphological variation in size, shape, the surface structure difference. During the present investigation, honey sample were subjected to the pollen analysis in which 18 pollen types were identified according to their morphological character. The pollen morphology provides information about the taxonomic character responsible, the anemophilous and entomophilous, mode of pollination and type of honey, the unifloral and multifloral. Present study reports multifloral types of honey in all samples studied. The some pollen types Altanthera, Triodax, Cassia, Parthenium, etc. The conclusion: it is noted that the pollen morphological character can very well assist the melissopinology study. It had the better identification and classification of pollen type. During the present investigation in this qualitative analysis of honey, pollen was carried out. The honey sample was collected from different study sites: Bhallar, Lathi, Ganeshpur, Kundra, Besa, Shirpur, in also in Vani Tahsil of Yavatmal. From the study, it is known that the pollen analysis of honey provides the information about the quality and quantity of pollen taxa present in the study region. The pollen analytical studies was provide the information about the resources of bee. The analysis of honey sample from pollen spectrum besides the enabling to inform the botanical sources of honey and also known as it is geographical sources. From the pollen analysis of different sample, it is the observed that this region have the diversity of bee flora. There are the 18 pollen type identified from different families: the Fabaceae, Asteraceae, Amaranthraceae, Brassicaceae, Bignoniaceae also. From the quantitative analysis of honey, it was observed that the most of the pollen in present sample number 3 Shirpur and sample number 4 Besa and sample number 6 Purad, in also less amount of pollen was found to be a sample 1 Bhallar. From the pollen analysis of different honey sample observed that this the region have diversity of bee flora. The qualitative analysis of honey, it consult that the majority of pollen load it is found to be from family Fabaceae and Asteraceae. Scope of this study: in this topic for the in general. Through the melisopynological study, we can compact fraud and incorrect labeling of honey. The information gained from the study of a given sample of honey, the total amount of pollen and their frequency can be calculated. Quantitative and qualitative analysis in this method. It is my references, and thank you so much